Hello, my name is Marina Driver, and my project is on the impact of media nutrition on microaggregate formation of Batrachochytrium dendrobatidis, also known as BD. BD is a global pathogen that infects and kills amphibians, and it was first discovered about 20 years ago by Dr. Joyce Longcore, and her work is what has led to many researchers worldwide investigating how to prevent BD from further infecting amphibian populations in our communities. BD belongs to Chytridium mycota, and it's characterized by having a wall-less uniflagellated motile stage in its life cycle and a reproductive stage, which is a chitin-walled zoosporangium. BD colonizes the skin of frogs by colonizing the keratinized mouth part of tadpoles and the skin of adult frogs. So BD's impact on the body, the main takeaway from this is that it interferes with normal skin functions. The skin is one of the three modes of respiration for frogs. So when BD causes hyperkeratosis, which is the thickening of the epidermal layer, as you see on the belly of this frog here, it prevents proper breathing from occurring. In addition, it also interferes with sodium-potassium transport. This interferes with the nervous system in general of the frog. So it causes the loss of the riding reflex, which is the ability of the frog to go from on its back to right side up. So here we see in this photo down here, the frog has been essentially paralyzed, so it is stuck on its back and it is not able to flip over because of the loss of the riding reflex. And this eventually leads to cardiac arrest and death. So the life cycle is characterized by having two different phases. So the first phase is the motile zoospore phase, which has a flagellum, so that way it can swim around and initiate the colonization of the host in general. And from there, it goes to phase two, where it forms a sessile sporangia, which is characterized by having a chitin-containing walled thalli, rhizoids, which are many roots, a retracted flagellum, and zoospores, which develop inside of it until it is mature. So in this diagram here, we see that from phase one, it is embedded inside of the host cell, developing into that thallus. And then from there, it starts to mature. And inside of here, we can see the zoospores, which are developing. And when it is mature enough, this cap on top opens, so that way the zoospores can go out and either reinfect the host or go on to infect other hosts in the environment. So in vitro versus in vivo lifestyles are a bit different for BD. So in vitro, the sporangia will aggregate and form a film-like structure, which we can see here in the top right. It creates an extracellular matrix which contains sugars and possibly protein and DNA. And this basically shows that BD is an opportunistic pathogen, not an obligate one. So it's able to survive without a host in an abiotic environment. And then in vivo, it only occurs inside of the keratinized skin of the frog, which is either the mouth part of the tadpole or the skin of the adult frog. The zoospores will adhere to the skin, and then from there burrow inside and start the reproduction phase. Our current project is investigating the adaptation of BD from H broth, which is the nutrient-rich media that we use in the lab for pure cultures, to pond water, which is nutrient-poor media. Our hypothesis is that growth and microaggregate formation of BD in nutritionally poor media can be enhanced by the presence of a leaf in the diluted medium. This is because it would allow for the BD to adhere to something and just start the formation of our extracellular matrix sooner. So the dilution begins by taking a pure culture of BD, which is grown in our H broth, and slowly, week after week, diluting it down with pond water. So that way the BD is not shocked initially and it's able to slowly adjust over time to the nutrient difference between the two. The pond water we used in our experiment, there were 3 different types and they were all from ponds in the area that have not tested positive quite yet for BD. So it was just a way for us to see its potential to colonize the area outside of the one pond that has tested positive so far. So from here, we hope to do protein analysis to determine the nutritional effects on protein expression and in BD. This would also allow us to further study microaggregate formation in Lubbock ponds, which have tested positive for BD, just to gain a better understanding of how it came to be here in Lubbock, since it's only so far been found in one of the many lakes in the surrounding area, or ponds in the surrounding area. So from here, we will also be able to search for factors which may contribute to environmental susceptibility to the fungus. So this may include certain pesticides or pollutants in the area. And hopefully by identifying specific factors, we can try to do preventative measures by taking it out of the environments that could be susceptible to it at the moment to prevent BD from affecting the amphibian population in Lubbock further.